Chapter 13. Control of movement. Control of movement. In more advanced forms of animal life, reflexive motion is based on the transmission of impulses from a receptor through an afferent neuron and ganglion cell to motor neurons and muscles. This arrangement is found in the reflex arc of higher animals, including humans, in whom the spinal cord has further developed into a central regulating mechanism. Superimposed on these reflex circuits, the brain is concerned with the initiation and control of movement and the integration of complex motions. Control of movement in humans. The motor system in humans controls a complex neuromuscular network. Commands must be sent to many muscles, and multiple ipsilateral and contralateral joints must be stabilized. The motor system includes cortical and subcortical areas of gray matter, the corticobulbar, corticospinal, corticopontine, rubrospinal, reticulospinal, vestibulospinal, and tectospinal descending tracts, gray matter of the spinal cord, efferent nerves, and the cerebellum and basal ganglia, Figs 13:1 and 13:2. Feedback from sensory systems and cerebellar efferents further influences the motor system. Movement is organized in increasingly complex and hierarchical levels. Reflexes are controlled at the spinal or higher levels. Stereotypic repetitious movements, such as walking or swimming, are governed by neural networks that include the spinal cord, brain stem, and cerebellum. Walking movements can be elicited in experimental animals after transection of the upper brain stem, probably as a result of the presence of central pattern generators, or local circuits of neurons that can trigger simple repetitive motor activities, in the lower brain stem or spinal cord. Specific, goal-directed movements are initiated at the level of the cerebral cortex. Figure 13:1. Schematic illustration of some pathways controlling motor functions. Arrows denote descending pathways. Figure 13:2. A. Basal ganglia, major structures. MD, medial dorsal, VA, ventral anterior, VL, ventral lateral nuclei of thalamus. B. Major efferents to basal ganglia. C. Intrinsic connections. D. Efferent connections. Major motor systems. Corticospinal and corticobulbar tracts. A. Origin and composition. The fibers of the corticospinal and corticobulbar tracts arise from the sensory motor cortex around the central sulcus, see Fig 13:1, about 55% originate in the frontal lobe, areas 4 and 6, and about 35% arise from areas 3, 1, and 2 in the post-central gyrus of the parietal lobe, see Fig 10-11. About 10% of the fibers originate in other frontal or parietal areas. The axons arising from the large pyramidal cells in layer V, Bitz's cells, of area 4 contribute only about 5% of the fibers of the corticospinal tract and its pyramidal portion. The portion of the pyramidal tract that arises from the frontal lobe is concerned with motor function. The portion from the parietal lobe deals more with modulation of the ascending systems. The tracts have endings or collaterals that synapse in the thalamus, ventral nuclei, the brain stem, pontine nuclei, reticular formation, and nuclei of cranial nerves, and the spinal cord, anterior horn motor neurons and interneurons, Fig 13-3. A direct pathway to spinal cord motor neurons exists only for the musculature of the distal extremity, such as the fingers that require rapid and precise control. B. Pathways. The corticobulbar, corticonuclear, fibers originate in the region of the sensory motor cortex, where the face is represented, see Figs 10-13 and 10-14. They pass through the posterior limb of the internal capsule and the middle portion of the crus cerebri to their targets, the somatic and brachial efferent nuclei in the brain stem. The corticospinal tract originates in the remainder of the sensory motor cortex and other cortical areas. It follows a similar trajectory through the brainstem and then passes through the pyramids of the medulla, hence, the name pyramidal tract, decussates, and descends in the lateral column of the spinal cord, see Figs 5 to 13, 13 to 1, and 13 to 3. About 10% of the pyramidal tract does not cross in the pyramidal decussation but descends in the anterior column of the spinal cord. These fibers decussate at lower cord levels, close to their destination. In addition, up to 3% of the descending fibers in the lateral corticospinal tract are uncrossed. These ipsilateral descending projections control musculature of the trunk and proximal limbs and thus participate in the maintenance of an upright stance and in gross positioning of the limbs. The pyramidal tract has a somatotopic organization throughout its course. The origin, termination, and function of this tract have been described more fully in Chapter 5. Figure 13-3 diagram of the corticospinal tract, including descending fibers that provide sensory modulation to thalamus, dorsal column nuclei, and dorsal horn. The extrapyramidal motor system. The extrapyramidal system is a set of subcortical circuits and pathways, phylogenetically older than the corticospinal system, which includes the corpus striatum, caudate nucleus, putamen, and globus pallidus, together with the subthalamic nucleus, substantia nigra, red nucleus, and brainstem reticular formation, Figs 13-2a, 13-4, and 13-5. Some authorities include descending spinal cord tracts other than the corticospinal tracts, such as the vestibulospinal, rubrospinal, tectospinal, and reticulospinal tracts, in the extrapyramidal motor system. Cortical and subcortical components of the motor system are richly interconnected, either directly and reciprocally, or by way of fiber loops that involve the extrapyramidal system, and the majority traverse the basal ganglia. Figure 13-4. Magnetic resonance image of a coronal section through the head at the level of the lentiform nucleus. Figure 13-5. Magnetic resonance image of an axial section through the head at the level of the lentiform nucleus. Basal ganglia pathways and nuclei. The anatomy of the gray masses in the forebrain that make up the basal ganglia has been described in Chapter 10, Fig 13-2. The striatum, caudate and putamen, is the major site of input to the basal ganglia, see Fig 13-2b. The striatum receives afferents via the corticostriate projections from a large portion of the cerebral cortex, especially the sensory motor cortex, areas 4, 1, 2, and 3, the more anterior premotor cortex, area 6, and the frontal eye fields, area 8, in the frontal and parietal lobes. These corticostriate projections are excitatory. The striatum also receives inputs from the intralaminar thalamic nuclei, substantia nigra, amygdala, hippocampus and midbrain raphe nuclei. Many inhibitory, gamma-aminobutyric acid, GABAergic, and a smaller number of excitatory interneurons, the latter in some cases using acetylcholine as a transmitter, are present within the striatum. The caudate and putamen send inhibitory, GABAergic, axons to the inner part of globus pallidus, GP, which is the major outflow nucleus of the basal ganglia. These projections provide a strong inhibitory input to the globus pallidus, see Fig 13-2c. The globus pallidus, GP, internal part, is one of the two major output nuclei of the basal ganglia. GP sends inhibitory axons, GABAergic, to the ventral nuclei, ventral anterior, VA, and ventral lateral, VL, of the thalamus, which also receives input from the cerebellum, the subthalamic nucleus, and substantia nigra. Axons from the globus pallidus project to the thalamus by passing through or around the internal capsule. They then travel in small bundles, the ansa lenticularis and the lenticular fasciculus, also known as the H2 field of Forel, before entering the thalamus, see Fig 13-2c. The VA and VL thalamic nuclei complete the feedback circuit by sending axons back to the cerebral cortex, see Fig 13-2d. The circuit thus traverses, in order: cortex sends to, striatum sends to, globus pallidus, internal, GP sends to, thalamus sends to, cortex. Another important feedback loop involves the second major outflow nucleus of the basal ganglia, the substantia nigra, which is reciprocally connected with the putamen and caudate nucleus. Dopaminergic neurons in the pars compacta of the substantia nigra project to the striatum, the nigrostriatal projection, where they form inhibitory synapses on striatal neurons that have D2 dopamine receptors, and excitatory synapses on neurons that have D1 dopamine receptors, see Fig 13-2b. Reciprocal projections travel from the striatum to the substantia nigra, striatonigral projection, and are also inhibitory, see Fig 13-2c. This loop travels along the pathway cortex sends to, striatum sends to, substantia nigra sends to, striatum. Neurons in the substantia nigra and GP also send inhibitory projections to the thalamus, VA and VL, which, in turn, sends projections to the sensory motor cortex. Substantia nigra, pars compacta, also sends modulatory projections, mesolimbic and mesocortical projections, to the limbic system and cortex. This pathway involves the following circuit. The pars reticulatum of the substantia nigra, SNR, receives input from the striatum, and sends axons outside the basal ganglia to modulate head and eye movements. The subthalamic nucleus, also called the nucleus of Lahaye, also receives inhibitory inputs from the globus pallidus and from the cortex. Efferents from the subthalamic nucleus return to the globus pallidus, see Fig 13-2c. Thus, the subthalamic nucleus participates in the feedback loop: cortex sends to, globus pallidus sends to, subthalamic nuclei sends to, globus pallidus sends to, cortex. Another loop involves the cerebellum. Portions of the thalamus project by way of the central tegmental tract to the inferior olivary nucleus. This nucleus, in turn, sends fibers to the contralateral cerebellar cortex. From the cerebellum, the loop to the thalamus is closed via the dentate and contralateral red nuclei. Although there are no direct projections from the caudate nucleus, putamen, or globus pallidus to the spinal cord, the subthalamic